Hey guys! In this video series, we'll be going over the defining characteristics of cancer, from how it develops to what makes it so lethal, to further understand how current developments in cancer research aim to target and defend against cancer. In today's video, we will first begin by understanding what cancer is and how it develops. Cancer can be defined as the uncontrolled growth of abnormal cells. Such growth leads to detrimental forms of tumors, which are collections of these multiplied cells that create swelling in parts of the body. These tumors can be malignant or benign, with the first type referring to masses of cells that are able to invade neighboring tissues in a process known as metastasis. While benign tumors are those that are unable to invade and thereby do not metastasize. While there is no such thing as a good tumor, benign tumors, while sometimes painful and potentially dangerous, do not pose the same threat as malignant tumors. However, a benign tumor can become malignant and metastasize once the tumor begins to invade other organs and nearby tissues. We can classify cancers by the type of tissue in which they originate and form such tumors. The 5 major classifications are carcinomas, which originate in the skin, lungs, pancreas, and other organs and glands. Sarcomas, which arise in bone, muscle, fat, blood vessels, cartilage, or other soft or connective tissue of the body. Melanomas, which occur in the cells that make the pigment in skin. Lymphomas, which arise in lymphocytes, the infection-fighting cells of the immune system. And leukemia, cancer of the body's blood-forming tissues, including the bone, marrow, and the lymphatic system. But how do such cancers occur? Well, it all has to do with the cell cycle. The cell cycle is the process in which cells divide. In normal cells, the cell cycle is controlled by a complex series of signaling pathways by which a cell grows, replicates its DNA, and divides. This process also includes mechanisms to ensure errors are corrected. And if not, the cells undergo programmed cell death, or apoptosis. In cancer, as a result of sustained genetic mutations over a lifetime, this regulatory process malfunctions, resulting in uncontrolled cell proliferation. The cell cycle can be divided into 3 main parts: interphase, mitosis or M phase, and cytokinesis, with each phase having their own series of steps. The key checkpoints in cell division occur during interphase and mitosis, with the G1, G2, and M checkpoints. Since G1 is the first step in interphase, where the cell grows before replicating its DNA, this checkpoint determines whether or not the cell should even replicate the DNA. At this checkpoint, the cell is checked for cell size, adequate nutrients and growth factors, and DNA damage. Cells with damaged DNA or a deficit of other resources are arrested and undergo apoptosis, or programmed cell death. A second such checkpoint occurs at the G2 phase, following the synthesis of DNA in S phase, but before cell division in M phase. Here, the cell checks for adequate cell size once more, but also ensures that the DNA replicated in S phase does not contain any major mutations that might otherwise harm the cell during mitosis. Again, if this check is not met, then the cell undergoes apoptosis. The final checkpoint occurs during the M phase, or mitosis, when the cell is undergoing metaphase. Here, the cell ensures that the spindle fibers are attached correctly to the chromosomes. This is important because the spindle fibers ensure that the chromosomes are able to separate evenly into the 2 new daughter cells made during mitosis, so that each new cell has the same amount of genetic information. A mistake made during this process is known as non-disjunction, which can lead to cancer cells due to the production of abnormal number of chromosomes in a patient's cells. While different types of cancer cells involve different types of mutations in the cell cycle, 2 types of cell cycle regulators promote the development of cancer: overactivated positive regulators and inactivated negative regulators. Positive regulators include the overactive cancer-promoting genes called oncogenes and the normal, not yet mutated forms proto-oncogenes. Many of the proteins that transmit growth factor signals during the cell cycle are encoded by proto-oncogenes. Normally, proteins drive cell cycle progression only when such growth factors are available. However, if one of these proteins becomes overactive due to mutations that create oncogenes, such a protein could transmit signals even when no growth factor is around, causing unregulated progression of the cell cycle. Negative regulators include tumor suppressor genes, which are almost non-functional in a cancer cell. Tumor suppressors prevent the formation of cancerous tumors when they are working correctly. However, if mutated, they can affect proteins that normally halt cell cycle progression. An example of such a protein includes the p53 protein, which has three main jobs. It first halts the cell cycle at the G1 checkpoint by triggering production of a cell cycle inhibitor. This break allows p53's second job of DNA repair, which activates DNA repair enzymes. If the damage is fixed, p53 will release the cell, allowing it to continue through the cell cycle. If the damage is not fixable, p53 will play its final role, triggering apoptosis so the damaged DNA is not passed on. In cancer cells, p53 is often missing, non-functional, or less active than normal. Over generations, cells with faulty p53 tend to accumulate mutations, some of which may turn proto-oncogenes to oncogenes or inactivate other tumor suppressors. I hope this video helped you to learn more about what is cancer and how it develops.